I made a video talking about bacteria some time ago, and I briefly touched upon antibiotic resistance, so I thought I should expand a little on that topic. Antibiotics are medicines used to prevent and treat bacterial infections. Antibiotic resistance occurs when bacteria change in response to the use of these medications. These bacteria, dubbed superbugs, are not only harder to treat, they appear to be fitter in general, meaning they survive better in the host and cause more deadly infections. A study found that mice infected with antibiotic-resistant strains of Pseudomonas aeruginosa, bacteria that cause lung infections, were more likely to die without any type of treatment during the study period than mice infected with Pseudomonas aeruginosa strains that did not have antibiotic resistance. The antibiotic-resistant strains were also better able to kill certain immune cells. Antibiotic resistance is on the rise to dangerously high levels all over the globe, and it's accelerated by the misuse and overuse of antibiotics, as well as poor infection prevention and control. Usually, when infectious bacteria mutate in a certain way and then multiply, they can become resistant to even the most powerful drugs. However, research has revealed a terrifying alternative way that antibiotic resistance can spread: an organism that passes on its resistance to other living bacteria. It all started when a patient had contracted the well-known superbug MRSA, methicillin-resistant Staphylococcus aureus. The medical term turned to one of the, quote, last line of defense antibiotics, the almighty vancomycin. This strain of MRSA originally had no natural defense against vancomycin. Roughly 2 months later, it had become resistant, rendering the treatment ineffective. Scientists would later uncover that rather than acquiring resistance through a simple mutation, the MRSA had instead been gifted a huge chunk of new DNA. Within this string of donated genetic code were the instructions for proteins that would keep the bacteria safe from the destructive work of the antibiotic. MRSA had been dealt a winning hand, but where had this DNA come from? From a traitor, living quietly inside our bodies. This little asshole is called Enterococcus faecalis, a bug that is typically described as a commensal bacterium, one of our so-called, quote, good bacteria, which lives happily in our guts, supposedly causing no harm. When bacteria come together, they can exchange information through DNA-encoded instructions. This is known as horizontal gene transfer, where copies of DNA move from one cell to another. Unfortunately, Enterococcus faecalis and its superbug compatriots have all the best information to share, information that allows them to survive antibiotics. And faecalis has gone a step further on its evolutionary journey, becoming one of the ultimate dealers of antibiotic resistance. One defense mechanism used by bacteria to guard themselves against unwanted genetic code is the CRISPR-Cas9 system, which scientists are also now using as a way to edit DNA. The system originated as a means for bacteria to chop viral DNA and other potentially dangerous genetic code to pieces before it caused them harm. Enterococcus faecalis once harbored the important CRISPR-Cas9 system, but, amazingly, sacrificed the defense mechanism so that all manner of DNA could enter and remain within the walls of the cell. This was a risky strategy, but ultimately proved worthwhile, unlocking the means for faecalis to acquire and subsequently pass on swaths of genetic knowledge. It was through this gain and exchange design that faecalis bestowed vancomycin resistance upon MRSA.